P19 cell line is known to differentiate into neurons. However, protocols for neuronal differentiation of P19 cell line are sometimes complicated. This method allows us to perform the neurogenic experiment in a user-friendly manner and expand the possibility of the usage of P19 cell line for the future.
To begin, maintain P19 cells in maintenance medium comprised of DMEM with 4.5 grams per liter of glucose, supplemented with 10% fetal bovine serum, 100 units per milliliter penicillin, and 100 units per milliliter streptomycin. Incubate the cell culture in a 37 degrees Celsius and 5% carbon dioxide environment until the cells are approximately 80% confluent. Remove the spent medium from the cell culture flask and wash the cells with 2 milliliters of calcium and magnesium-free PBS. Add 1 milliliter of 0.25% trypsin EDTA to completely cover the monolayer of the cells, and incubate at 37 degrees Celsius and 5% carbon dioxide for 2 to 3 minutes. Under a microscope, check to see if all the cells are detached. Resuspend the cells in 9 milliliters of maintenance medium in order to neutralize the trypsin. Transfer the cells into a 15 milliliter tube and centrifuge at 200 times G and room temperature for 5 minutes to pellet the cells. Then, aspirate the supernatant without disturbing the pellet. Resuspend the pellet in 10 milliliters of fresh maintenance medium and use a cell counter to determine the cell number according to the manufacturer's instructions. Seed 20,000 cells per